Hi everyone! Thanks for joining my webinar presentation today. I'd like to start off by introducing myself. My name is Jennifer and I'm a scientific sales executive with Rapid Novor. My background is in pharmaceutical chemistry, vaccine development, and mass spec. If you have any questions throughout the presentation, type them below and I'm more than happy to answer them at the end. If I don't have time to get to all of them, I'll make sure to follow up via email. So today I'll be going through the details of our de novo protein sequencing platform that we offer here at Rapid Novor. This will include information on how we have developed a service that obtains 100% coverage and accuracy, the process, why researchers use us, and finally, how you can sequence your antibody or protein with us. So let's get into it. Antibody research has been one of the fastest growing research sectors in the past decade. The estimated market value of monoclonal antibody therapeutics is expected to reach $300 billion in 2025. So many different applications of antibodies have contributed to the success of these important therapeutics reaching the market, including drug discovery, in vitro diagnostics, antibody engineering, CAR T therapy, and more. The problem is that antibodies don't always behave as expected. If you work with antibodies, you've most likely experienced the impact of the reproducibility crisis by now, even unknowingly. The surge in antibody development has led to a global antibody reagents market that is now worth $1.6 billion. We've seen a huge increase in biotech companies offering antibody reagents, immunoassays, and more. Because of the growth in the market, acquisitions, and bulk catalog purchases, many antibodies are being sold without having been rigorously validated. For example, some antibody suppliers sell the same reagents to different kit manufacturing companies. The result here is seemingly different ELISA kits based on the same antibody, making the selection process ever more confusing for researchers. Perhaps the biggest shocker is that according to multiple studies, on average, only 50% of antibodies bind to their intended target. Chances are, if you've faced issues with unexpected biological activity, inconsistent batches, or antibody supply chain hurdles, you've already been impacted by the reproducibility crisis in terms of lost research and money. Well, you're not alone. For example, these researchers published a paper based on their experience, which I've cited below. Dr. Diamandis and Dr. Prassas are translational researchers at the Mount Sinai Hospital at the University of Toronto. They bought an ELISA kit designed to quantify the protein CUZD1 and spent 2 years and over $500,000 to identify that the kit was actually measuring a completely different protein, CA125. Aside from all the time and money wasted, there was the rejection of probably promising biomarkers. In the end, everything needed to be re-examined by alternative technologies. Just imagine how devastating this must have been. The worst part is, this happens to researchers a lot more than you hear about it. So antibody reproducibility is becoming a growing concern for the life sciences industry, and for good reason. An estimated $350 million is wasted on incorrectly validated antibodies each year in the United States alone. The scientific community is starting to urge large organizations for government intervention. However, scientists are safeguarding their research efforts themselves. One way to prevent becoming a reproducibility crisis statistic is through securing your efforts through protein sequencing. So now let me give you an introduction to Rapid Novor, and I'll lead you into how we perform our amazing service from here. Rapid Novor is a protein sequencing CRO based in Kitchener, Canada. We're about a 1-hour drive from Toronto. Our president and CSO, Dr. Bin Ma, is an algorithms professor at the University of Waterloo and is world-renowned in the bioinformatics field. Dr. Ma previously developed a widely known and adopted commercial proteomics software. Building on that knowledge, he wrote a new program that incorporates machine learning and AI to exclusively focus on the de novo protein sequencing of antibodies, which is named REMAB. Broadly speaking, across the protein sequencing industry, the expectation is 200 micrograms of starting material, and most companies will only work with mouse antibodies. It's rare to see anything other than the purest proteins being accepted for sequencing, and turnaround times tend to be extremely lengthy. Rapid Novor has really revolutionized the industry and continues to bypass barriers that prevent most from attempting this. Our minimum sample requirement is only 100 micrograms, and we can handle any species, any isotype, since we are a fully de novo platform. We also now accept oligoclonal mixtures of antibodies. For example, a sample containing 5 monoclonal antibodies. Additionally, we frequently handle contaminated samples that other labs would reject. We have built a robust, high-throughput process that can fully de novo sequence an antibody in only a few days. Our standard timeline is 2 weeks for one antibody or 3 weeks for up to 10 antibodies. Now, I can go around and tell you about how we're faster and better, but I want to give you some examples of why we are. So, last year we made a new record for smallest amount of antibody sequenced. With only 9 micrograms of starting material, we pulled out a full and correct sequence. This project came from a Harvard researcher whose cell lines were unfortunately thrown away, and this is all they had left. The industry average for protein sequencing is 100 to 200 micrograms of starting material, so understandably, many companies would have rejected this project. However, at Rapid Novor, we welcome challenges and are willing to push our experimental procedures because we know that you don't want to lose your research, and we don't want that either. This is why we are able to take on this project and ultimately deliver the full amino acid sequence with full coverage, 100% accuracy, and allow the scientists to confidently proceed with their research. Now, when we're talking about speed, we must live up to our name, Rapid. We've built a robust, repeatable service that is unmatched. We've really sequenced more antibodies than anyone else. We did 650 in 2019 alone and have thousands more under our belt. Another note is that we can now handle oligoclonal mixtures, such as a mixture of 5 monoclonal antibodies, and we have a proven track record of being able to correctly match the heavy and light chains. I do want to take a minute and emphasize that protein sequencing is extremely difficult without the right expertise. Even to experienced proteomics specialists, protein sequencing proves to be a difficult task, taking them months to years, often with incorrect sequences or abandoned projects. It's due to our bioinformatics expertise and proprietary algorithms that we are able to accurately and quickly deliver full coverage sequences every day. This is why 18 of the top 20 pharmas outsource their protein sequencing needs to us. Peptide mapping used to be a solution before de novo sequencing became so accessible. However, for most standards, it is not suitable and it will not always be 100% accurate. If peptide mapping sees a combination not in the database, it can only guess, whereas our de novo approach can determine every amino acid with 100% confidence. Peptide mapping cannot differentiate between isobaric residues like isoleucine and leucine, but also isobaric combinations. For example, a glutamine looks the same as an alanine and glycine beside each other on the mass spec. So since our platform is fully de novo, we can guarantee 100% accuracy and 100% coverage on any species and any isotype. If you're not familiar with protein sequencing, that's okay. We're going to go through how everything works today. The basis of protein sequencing really comes down to obtaining the amino acid sequence. What we have done is developed a robust and repeatable platform using mass spectrometry and proprietary algorithms to de novo sequence proteins. I'll show you Rapid Novor's process now so you can see how we achieve such legendary science. Our system can be broken down into 4 steps: enzymatic digestions, LC-MS acquisitions using our thermo-orbitrap fusions, sequence assembly by our proprietary algorithm called REMAB, and expert reviews by our team of bioinformaticians. Before we start digestions, we always run a reducing SDS page gel to check on the purity. This gel will separate the heavy and light chains. Ideally, we see 2 perfect bands like the one on the left. However, we often see samples with contamination, such as the proteins like BSA, like the picture on the right. Now, most labs wouldn't think twice about rejecting a sample like this, but honestly, this is just another day in the lab for us, and we welcome these challenges. This is where the screening process is important for us. When we see contaminated samples, we can purify the antibody before digesting. Once the sample is ready to go forward, we do some chemistry to separate the heavy and light chains and cleave glycans. The material then gets digested by different enzymes, which will cleave the protein at very different positions along the amino acid chain. We've chosen the enzymes carefully so that they have very different cutting rules to increase digestion site diversity. Next, the digestions are run on our LC-MS systems to generate peptide datasets. This will be on one of our thermo-orbitrap fusions or our thermo-orbitrap fusion Lumos systems. Our Novor engine interprets the MS data and converts it into peptide sequences. The REMAB assembly algorithm assembles the peptides into the final protein sequence. For proteomics scientists in the past, isobaric residues have been a challenge. Isoleucine and leucine have the same chemical composition and molecular weight, and therefore look the same on the mass spec at first glance. These amino acids are of course found all throughout the antibody structure, including the CDR regions. Thus, being able to distinguish the two is crucial. We have invented an additional feature of our algorithm called WILD. This stands for W-ion isoleucine leucine determination. When we look at the mass spec data, isoleucine and leucine have the same Z ions. However, their W ions are different. With this W ion method, our software determines which is which, so you can be 100% confident in your sequence. We were the first to offer a commercial solution for distinguishing isoleucine and leucine, and most labs can't even do this. But we know that if we are off by just one amino acid, it could affect your protein's binding. Final step of our process is sequence review by our bioinformatics team. This ensures full and accurate coverage so that every amino acid is accounted for. We don't just settle for 100% coverage. Our team ensures there is enough overlap to promise 100% accuracy as well. Once the report is complete, we deliver it right to your inbox via a secure portal. The report contains the sequence in both FASTA and CHOTHEA scheme, notable observations and PTM insights, summary of procedures, coverage overview, and a breakdown of every isoleucine and leucine determination, as well as our interactive coverage viewer. Now, that's our Remab protein sequencing service in its entirety. We can get all this work done in just 2 weeks for one antibody. That's our service at a glance, but why do researchers choose us? It really comes down to our experience, accuracy, speed, and customer service. Rapid Novor has a team with well over 20 years of experience in the field. We've delivered over 1,500 sequences and 650 just in 2019. In September 2019, we moved into a brand new custom facility that has allowed us to expand our lab and seamlessly integrate our entire team under one roof. This allows us to efficiently meet timelines and, most importantly, keep your data secure. We are proud to say that we are the largest proteomics facility in Canada. Because we have focused on building a robust, repeatable, and high-throughput service, we proudly have the fastest bulk turnaround time. Due to this, our 100% accuracy and full coverage service is why we are trusted by 18 of the top 20 pharmas. DNA sequencing is another available commercial option that has existed as a commercial solution much longer than protein sequencing and is quite widely available. However, DNA sequencing requires access to the cell line, or DNA of course, and cannot detect PTMs. And PTMs can affect the overall binding of the antibody, which is a reason why some researchers do prefer protein sequencing depending on their use case. Protein sequencing detects PTMs, additional light chains, and glycosylations that may be present. Additionally, we detect and sequence other antibodies present, and we indeed have received samples from customers who had no idea there was more than one antibody in their sample. And of course, if you do not have access to the DNA or cell line, protein sequencing is the way to go since we only need a protein sample. With mass spec-based protein sequencing, we will tell you exactly what's in the vial that you send. So, if you have the DNA sequence and are still seeing unexpected biological activity, there's a chance that an unnoticed glycosylation or secondary light chain is present. This is where protein sequencing offers complete peace of mind. On the topic of other moieties lurking within your antibody, did you know that 10 to 15% of hybridoma-generated antibodies have an additional glycosylation? We've had a customer send us an antibody that they previously DNA sequenced but wasn't producing recombinantly. We found an additional glycosylation that the DNA sequencing could not detect. Also, a 2018 study found that 15% of antibodies do have a secondary light chain, so there's a chance that your antibody contains this. We often detect smaller amount of other antibodies, but more and more we are seeing supposedly monoclonal samples containing 2 or more full antibodies. We have a successful record of correctly sequencing these mixtures of 2 to 5 monoclonal antibodies. We've seen all kinds of different researchers using our service for many purposes. Some people want to engineer their antibody into a better therapeutic candidate, while others are sequencing scFVs for things like CAR-T construction. Others may need the sequence to recover antibody supply from a dead cell line, or simply need it for a publication. A big area that we're used for is securing reproducibility. Don't wait until batch variability ruins your research. Recombinantly express the antibody and immortalize your reagent. So if you'd like to get started, here's what we need you to gather. 100 micrograms of antibody or protein. Ensure that the sample is at least 80% pure. We typically estimate this on an electrophoresis gel. Ensure the sample is free of excessive protein contaminants such as BSA. What we can promise, 100% accuracy and coverage in 2 weeks for one antibody. Our system is built for high throughput, so we can also handle bulk orders easily. Our timeline for 2 to 10 samples is just 3 weeks total. So, why choose Rapid Novor? It's simple. We've sequenced more antibodies and proteins than anyone else in the world. This, along with our security, speed, and ability to handle any species and any isotype, are just a few reasons why scientists continue to use us and recommend our service. To contact us, my information is on this webinar page, or fill in our submission form on our website by clicking Ask an Expert. If you're looking to do some antibody engineering, secure your reproducibility, recover lost information, or learn more about how protein sequencing can accelerate your research, contact us and we'd love to chat.